Thank you for joining us. I'm Heather Gatney, and I'm the coordinator of the CVFS webinar series. Our host today is Coulter Mitchell, Associate Research Professor at the Survey Research Center at ISR. Coulter is going to give us an overview of the methodology around the construction of polygenic scores and their applications in demographic research. There will be a Q&A session after the presentation. I think you're also, Coulter, is it okay if people ask questions? I think you were fine with that last time.
Yeah, that's fine.
Feel free to ask questions during the presentation. We will have that Q&A session afterwards. You can always submit a question at any time today using the chat feature. We'd like to remind you that this webinar is being recorded. If you have any objections, you may disconnect at this point. A recording of the webinar and the slides will soon be available on the CVFS website. It's all yours, Coulter.
Perfect. Thanks, Heather. And Heather, if someone submits a question and you think it's something that needs to interrupt me, please feel free to do that.
Sure.
All right, everyone. Nice to see you and have you here. So what I'm going to try to do here is give you a 35-minute primer on polygenic scores. I can't promise you're going to be experts at the end of this, but you'll know a little bit. Hopefully enough that you can take a next step or decide that you never want to take a next step, either one. So I'm going to cover kind of what the potential is, and that's where you get kind of this, dream cloud, pie in the sky sort of idea of what's there. Why should we have genetic measures? Why are they important? Why, if I'm a social scientist, why would I be doing it here? Why would I study demographic research related to biology? Some of the pitfalls in how kind of this concept actually gets operationalized into a polygenic score. And then some of the practicalities, like really what's possible right now, and where's this likely going in the future? Okay, so just so you all know, when I'm thinking about biology, that covers lots of different things. That's far too big to discuss. So we're going to focus really on genes in this case, polygenic scores. Okay, so now we've made this a little more concise. So what's the potential? Let's just imagine that we can create a perfect measure of genetics, right? So here's some of the cool things that you get with genetics. It's constant. So you measure it at age 80, you measure it at birth, it stays the same, right? So you can actually do, you can collect something at the end of a cohort study and go back and redo all of your analyses using genetics. Pretty amazing, right? Nice, no retrospective recall, nothing like that. So remember, we have two pieces of information that come from one of each parent. That's important, but they don't change. And they're the same for all the different cells of your body. That's true for almost nothing else. So we have these cells that, whether or not you're collecting saliva or you're collecting blood, exact same DNA, makes it very, very easy to collect. Okay, it's predictive. So we talk about heritability estimates, and we can't really go into too much detail. There's some citations if you want to read a lot more, but these have used thousands of different behavioral measures and even non-behavioral measures for people who are doing that kind of work, medical sorts of studies, tens of millions of individuals using twins and parent-child and different types of sibling relationships. And they all show that genes are involved in behavior and to a pretty significant degree. Whether that's a direct effect or a gene environment interaction, they all kind of gets muddled in. That's part of the way that this gets estimated. But we're talking about from a scale from 0 to 1, somewhere in the vicinity of 50% on average of behavior is somehow connected to genetics. So what does that mean? That's a little more complicated. But just to give you a sense of what this looks like, fertility, number of children that you have, for example, anywhere is a range from 25% to 30% due to some sort of genetic component. And again, that could be direct genetic effects, gene-gene environment, gene interactions and gene environment interactions are all kind of included in that. Educational attainment is from 25% to 35%, depression, 40% to 60%, alcohol use. And you can do this for just about everything. Almost everything is somewhat heritable. One thing to know about heritability, and this comes back many, many times, is that's a population level measure. It's not that every single person's behavior, like what you're eating today, is somehow 30% genetic. That's not what we're talking about. This is on average at a population level. Demographers use that all the time. This is the same is true for genetics. And there's lots of assumptions. Again, we can talk about that another time. But that's kind of the potential. The other thing is especially over the last few years, it's relatively inexpensive to collect these. So imagine trying to do a survey where you can get millions of variables for less than $100 per person. That's almost impossible to pull off. So you get a lot of information for relatively little cost. So why should social scientists even care about this in general? I've said this in another presentation, but one way to think about it is, even if you don't care at all about the genetics, it's better measurement of this of kind of social contextual effects. If you can get rid of anything that's due to genetics that's not due to the environment, it improves your controls. So it improves your power to detect the social effects, increases the strength of your argument by removing some potential confounders, because there will always be people who say, well, it's not really about social things, it's really about genetics. You can control for that. And again, don't have time to get into this. There's some citations down below, but you can potentially use genetics as an instrument, so an exogenous shock, because there are some random aspects to which allele, which genetic version you get from which parent and how those all combine. There is some randomness in there, and people use that to have essentially an exogenous shock. So there's some interesting things you can do. Okay. The other thing that social scientists care tremendously about is moderation. Why is it that given all the same kind of characteristics, people have dramatically different responses to, to any kind of social context or, a trial or something like that? And again, a lot of work, people looking at gene environment interactions, that kind of moderation. Another thing that doesn't get as much press, but I actually think is really important, is gene environment correlation. So this isn't the interaction, just as why is it that we see consistent patterns of demographic and social sorting over geography and time, and actually even intergenerationally. And in that case, we have things like active, this is one theory of genes, or one aspect of genes, is there something about something innate within us that's kind of pushing us to choose certain environments? Or is it evocative? It actually is manifesting in some other way. And those around me are kind of selecting me into different things. So it's really the environment selecting people as opposed to you selecting it. So one way I often think about this is we often talk about children, and if children are responding a certain way and acting a certain way due to something innate within them, the parents might change their parenting techniques, something like that. But there's less obvious ways that's happening too. And passive is genes and environments are kind of passed down together. It's hard to disentangle. So there's a lot of work that could be quite interesting to do with this, that are genetics and environmental combined. So do the genes actually shape the environment? Well, let's go through a couple. Here's an example of one. I'll just show you a few things from a paper that came out of the Dunedin study by Dan Belsky, is the first author. And one of the things that they found is using polygenic scores, which we'll talk about more. So these genetic measures of educational attainment, that people with polygenic scores of educational attainment, remember, don't change over time, are more likely to measure people or to marry people who have similar polygenic scores. So there's, is it really genetics that are driving people to be together? Or is that selection of a different kind? And in fact, what you see is that it's also true that even within like low SES families, middle SES families, high SES families, there's still this kind of effect of the polygenic score, again, for education in this case, that's the x-axis, affecting or predicting adult educational attainment. And even the same score is predictive of whether or not you migrate out, in this case, out of New Zealand to other locations. So that's more of an active gene environment correlation story. So you can see genes are certainly correlated. How are they correlated is particularly interesting. And I mentioned this before, passive gene environment correlation and what we might call genetic nurturance. And that is that the genetics of the parent are clearly passed down to the child, and that might therefore influence college education. So that's the mother influencing the child's genes influencing college attendance. But in addition, the genes of the parent are also shaping the environment, maybe the cognitive stimulation, how much they're reading, their math ability, things like that. And so you can imagine several different kind of parenting level measures that are also influencing college education. So maybe genes are operating through these social mechanisms. And so again, kind of an exciting way to tease out certain processes that we already can document are having an effect. So one other reason why social scientists might want to care about this are just that it's a really nice way of highlighting the social environment. So it reveals the effect of an environment by comparing the strength of genetic effects. So let me show you an example using the Wisconsin Longitudinal Study. So at first they took, so Wisconsin Longitudinal Study is a cohort study, but it's a high school cohort study, and they're now, I think, in their 70s, 80s maybe. So anyway, they took their genetic measures, which they got much later in life, and they can go back and they look, okay, we create these polygenic scores. Again, in this case, it's a polygenic score for education. And I'll explain why a lot of these workers using the polygenic score for education. And you see like, oh, between men and women, men are yellow, yes, and women, female are blue. And in terms of high school rank and IQ measures in high school, the polygenic score really didn't distinguish between men and women, right? So it was pretty even prediction. And by the way, you wouldn't expect this to have a difference between men and women at all. It should be a null. But then you compare actual educational attainment of these same people at different time points, age 25, age 36, age 54, age 65, and you show the effect of the polygenic score on educational attainment at those different ages. And what do you see? That the effect of the polygenic score for men is significantly higher and O is higher, right? And actually kind of expands. Why might that be? Well, during this time, women were not getting as much education as men, and in some ways were constrained to do so. So therefore, their genetic effect won't be as large as men's because the environment is tamping down on that potential. So it's a way of highlighting the effect of social influences using what should be, in this case, a very similar effect, right? So that's a nice example of that. Okay, so just as a reminder, here's the exciting part, right? This is when it's perfect. It's the pie in the sky kind of dream scenario. These are consistent measures. They're predictive. They're pretty inexpensive, relatively speaking, useful for lots of different applications that I just showed you. So the question is, why isn't everybody on board with doing this? And that's where we get to the pitfalls. In order to talk about the pitfalls, I'm just going to talk a little bit about genetic research, a little bit about GWAS, the construction of polygenic scores, a little bit on ancestry, sample size, and selection. We spend like a whole day on this when we teach this course. That's a week-long course, Elvin. So you could spend a whole semester really just studying all these. So bear with me. We'll go through this. Hopefully you have some questions and some ideas when we come through with it. So just as a reminder, when we're measuring the genome, there are lots of different ways you can do it. Typically, what we're using are what are called single nucleotide polymorphisms. Those are those base pairs that you all remember from biology. That's the simplest form of genetic variation. Remember that most of our genome is actually identical across all humans. So SNPs really only occur 1 in every 300 base pairs. And what we require in order for it to be called a SNP, a single nucleotide polymorphism, is that there's some variation in at least 1% of the population. If it's less than that, it's too rare, and that becomes a different measure. And most of them have no variation. And so allele is the way we talk about variance, right? And remember, you get one from your father, one from your mother. And so you can either have 0 copies of that allele, 1 or 2. Does that make sense? So it's a really nice simple coding, 0, 1, or 2. So what do we know? What we know is that for years, we've been doing genetic work long before we could do genotyping. And we found a lot of the really rare, what we call Mendelian diseases, right? So that is where you have one small variation. It changes so much of your biology that you are definitely getting this disease. A lot of times you die really young. Sometimes you don't, but those are pretty rare. And we found those before we could do genotyping. So there are other ways that we find those things. And so it was much, much easier. What we found is that almost everything is actually, and that's this rare, this upper thing here with very large effect sizes, but very rare in the population. Again, that's that allele frequency down here. Remember how I said 0.01? That's down here around the low frequency. That's kind of our cutoff for what we're looking for. And what you see is that most of the genetic variation that we see in the world is driven by pretty, what we call common variants. So at least 1 to 5% of the population has that version. And that's driving most of the genetic variation. It's very rare to have really rare variants that have small effects. That's almost impossible to detect. And very large effects of common variants eventually would be selected out of the population through, natural forces. So anyway, keep that in mind. We're looking for lots of measures, lots of genetic variants combining to explain the genetic variation. So in order to do that, we now use what's called a genome-wide association study or genome-wide analysis. So I'm going to describe a little bit about how that works. And we've talked about this in the past. So typically we start with pretty simple models. We put an outcome that we want to explain. In this case, let's say depression, whether or not you're depressed or not, or a depressive scale, right? And what you do is you take all of these millions of genetic variants that we've now measured, up to like 25 or 30 million, and you run that many regressions, having a few controls, very small number of controls. And I'll show you what that looks like. In order to pull that off, though, you can't just do it in your study, right? That'd be too many tests for your data set. So we do it in what we call large meta-analyses or genetic consortium. And then we take the things that we find that are genome-wide significant, p-value of 5 × 10⁻⁸, and we try to replicate it in a sample that wasn't included in the discovery analysis. So this is completely agnostic to what you think is going to happen, the effect of any particular gene. You're not selecting anything. You're just letting the data tell you what's significant and what's not. And then you test it again, you replicate it in another study. So I think I've shown this before. You can pick this and I'll show you all the different EWASs that have existed to some degree. This is just one. And this is, if you look at this Manhattan plot, doesn't really matter what it's for. This is the address of the variant. So remember, we have all these different chromosomes down here. And you have different genetic variants along that chromosome. So it's literally an address. Each dot is one of those SNPs that we talked about before. And this is the -log10 for the p-value. So the higher you go, the more zeros there are. So, 0.01 or 0.05, like we normally think of, is way down here at the bottom. In order to be genome-wide significant, it has to get over these lines. And you start to see there are certain regions. And that's all GWAS was about, is locating regions of the genome where something seems to be correlated with the outcome that you're interested in, right? And you look for these long trails and you see that that works well. So yeah, it's the strength of the association. So this is the basis of current genomic research, especially in humans, is just doing these. Thousands and thousands of these on every single outcome you can possibly imagine. So what are the strengths and the weaknesses of this? Well, the strength is you use a meta-analysis approach. You have, instead of having, the 10,000 people in the Chitwan Valley Family Study, you can have 500,000 or 1 million or 2 million because you combine across 50 or 100 different studies, right? And actually what you find is that the more things you put in, the more predictive the polygenic score will become that you'll create off of this. So, and all cohorts measure the same SNPs due to some other technical things I won't get into. And even more important than that, remember that they're all coded the exact same way, 0, 1, or 2, 0, 1, or 2 for all of these measures. I want you to imagine briefly that we did this in the social sciences. You could imagine having something like that, where let's say you have some measure of health or well-being that you really care about, and you use all of the survey data that you've ever collected, but you've happened to have collected all of the data so that it's measured as a 0, 1, or 2, right? For all of your questions, it's either, or maybe just 0, 1 or something like that. And then you just did essentially a regression of all of those different things. That's what we're doing here, except instead of every single survey question, it's every single genetic marker. And that also might help you realize what some of the problems are and why molecular biologists also have issues with this. Because you might care more about one type of question or one type of, or one region of interest. So here's some of the limitations. The measurement of all of the traits, so that outcome that we, varies by cohort. Imagine just trying to combine all the US studies or all the South Asian studies, and how slight differences in language or slight differences in the exact measure that they use all could change the interpretation of what it means to be depressed, for example. It also assumes that there's a common genetic effect regardless of differences in non-genetic factors across cohorts, SES, age, all of those different things that we know probably do have different. And it obviously limits the complexity. That kind of goes back to the measurement. If you only have one point in time for most studies, you're not going to do a longitudinal analysis. So, so now we're getting into the meat of, you have a GWAS, right? So like I said, we literally have thousands and thousands of these GWASs, and they've all, a lot of them, been published in different places, and they often have to report their results. So what we do is, if you have a new study like CVFS, and you show up and you say, I want to construct a polygenic score based on one of these GWASs, you go to that, you pull off the results for, say, a depression GWAS, and then you, so that's, pretend that's the depression GWAS. What you do then is you take your genetic data, which again are coded 0, 1, or 2, you multiply it by something that's indicative of the strength of the association for that specific genetic measure. So that's SNP. So you take your SNP data, you multiply it by the weight. Usually it's the inverse of the p-value, or it's the effect size, something like that. And that ends up, you then sum across all the SNPs that way. So that what that results in is some of these genome-wide significant hits end up having very strong weights or very high weights, and the very low weights are these ones down here that are, have really no effect whatsoever. So that gives you a polygenic score for, let's say, depression for this new sample. And all it is a predictive algorithm that sorts people on their probability, not their probability necessarily, but the tendency to be higher or lower in depression. And the nice thing is, and that's essentially what we were talking about before, GWAS plus your own data, you start creating these scores. And the nice thing is if you just had one SNP, you would either be 0, 1, or 2, and you would have a not very normal distribution. But as you start adding more and more of these, you end up having this beautifully distributed, normally distributed measure that arrays your entire data set based on predicted, like depression, for example. There's a lot that goes into these scores, a lot of decisions. If you're really interested in it, you can learn more by going to this paper. Okay, so what are one of the other pitfalls that comes into this? So I just want to be clear, first of all, to clarify a couple things. One is often people will confuse the term race, ethnicity, caste, that's one set, versus ancestry, and they'll combine them all. Race, ethnicity, caste are socially constructed and are often used in determining social and health inequalities. Ancestry is a variation in genetic architecture between populations. So that's used in genomic analyses. I want to be clear though, that doesn't mean that like this, if you think about these population differences in genetic architecture, so these allele frequency differences, those are a result of things like migration, fertility, mortality, who gets to marry who gets to reproduce with whom, who dies faster, who dies slower. All those things end up shaping the genetic architecture, but that's what gets used. Genetic variation is not necessarily a causal effect with biology and health, but it can be often highly correlated. But the key thing here is that ancestry and race do not perfectly overlap. And in fact, for some race ethnic groups like Hispanics and in the United States, African American, the number of ancestries is actually can be quite large, and the variation within it is quite large. Whites tend to typically mostly be European ancestry, but that's kind of the exception in a lot of ways. A lot of the times there's a lot more diversity of ancestry within race. And why is that important? That ends up being important for one kind of simple thing, or two really, but one for now, and that is that the older a population is, due to the way genetics worked, and especially population genetics, there's this interesting thing of you have larger, the more similar you are to people and the newer your population, the larger the chunks of DNA you share with them. So if we want to go very specific, you look at like your parents and you clearly share big chunks of your DNA with your parent, with your mother and your father, right? So if you think about that on a population level, the newer a population, the fewer generations that have been there, the larger the chunks you share with them. So European ancestry are actually some of the newest ancestries. And so they actually end up sharing big chunks of their DNA. So it's much easier to find certain variants. And that's what this upper left-hand corner is. You have the same variant is correlated with lots of other genetic variants. So this top one is the causal variant, the one we think is associated with the disease. There's big chunks of other things associated with it. On the bottom right, in an older population, this is Yoruban, for example, in Africa, many, many more generations, the area in which they share with other people and in which you can find kind of correlated measures is much, much more narrow, if that makes sense. So it's the genetic variation in older populations. And by this case, I mean population like Yoruban versus European, not like age of someone. Anyway,
Coulter,
go ahead.
Quick question, Coulter. The way you just described those as older, younger, I'm trying to wrap my head around from the perspective of somebody who studies marriage and family.
Yes.
I hear that as young means a lot of intermarriage and old means a long time without intermarriage. Am I wrong?
No, I think that's right. So a different way to think about it is, think about it in terms of, there's just more time for your genome to essentially get divided up because you've had more generations. And because, if you think about let's imagine you were starting a whole new ancestry group. In fact, you do. That's what each family is a new ancestry group. It just hasn't been around long enough to be called that. Then yes, if you just keep staying within that same group, you'll share a lot of the genome with those people. And the ones who have had lots of generations have smaller ones. You can go into this in a lot of detail. This is population genetics. But the reason why this ends up becoming important is because the European ancestry is actually so much more homogeneous than every other ancestry group. It's much easier to do genetic research in that because you can find a lot more causal variants. You can find a lot more significant and correlated hits. it would take significantly more people to find the same kind of genetic power, statistical power that is, within a Yoruban population, because the genetic variation is so much higher there. So the reason why that's important is this next slide. And this is the whole point behind that, and probably too much detail for that, is to just say what ends up happening when you create polygenic scores is you end up not being able to compare across ancestry groups. So if you were to take this and look at height, for example, we'll just focus on that one. Here's the European ancestry, and it looks like they have higher polygenic scores for height. And on the far side is East Asian, and this red is African. So this would suggest that Africans have significantly lower probabilities of being tall, so they're much, much shorter than European Americans, for example, which is not true, right? That's simply not true. So it's because of these genetic architecture differences. If you kind of compare within or between ancestry groups, you can have a false sense of how genes are driving things that have nothing to do with the underlying biology. So that becomes really tricky. So you can really only look at things within ancestry groups, which is one of the drawbacks. Another key part to this is there's selection into GWAS, and this is a slightly different thing, but still related. The vast majority of GWAS results are based on only European ancestry for the reasons I was saying before. There's some ethnocentrism for sure. That's also where wealthier countries are typically located. And because it's so much easier to do genetic work within that ancestry group, almost all the GWAS, if you look at this bottom, the fraction, almost all GWASs are performed in European ancestry. And that does not match on to the world, right? So now you're starting to see there's some issues here with how portable these might be across ancestry groups. But the reality is, and this is a great paper if you want to look at it, that's not just true about ancestry. That's actually true about men and women, and it's true about age, and it's true about SES. Essentially, there's just selection into who participates in GWAS, and that affects who it potentially represents the best. In this case, what this is showing is, if we look at diastolic blood pressure, if you look at it, the polygenic score created in men and women combined, it looks like it's working better for women than it is for men. But then when you do a GWAS in just women or GWAS in just men, you start to see like, actually, this is all driven by the fact that there's some selection who's participating in this particular GWAS. And it's just not going to work well for men ever. And the same true for BMI, years of schooling. This paper really kind of changed the way we think about it. But for social scientists, you're used to this. If you start creating measures and kind of population estimates based on surveys that are biased, you will have biased estimates. And that's essentially what this is saying. The reason why this is important is ascertainment bias and assay bias. So that's who participates or how well your measures work for different groups, in this case, ancestry or different populations, leads to an algorithm bias. And that's the polygenic score, which then ultimately leads to an anchor or confirmation bias in the literature, because people think, well, this is the way this polygenic score works for everyone, when the reality is it was biased to begin with the GWAS. This is for people who want to look up more. There's lots of reviews on this. Okay, so some quick to close out, some practicalities of thinking about biology to polygenic scores, right? So I probably got your hopes up high, saying how great they were. And then at the end, I was kind of saying, here are all the reasons why this is deeply, deeply flawed. So what do I actually think is going to happen? So here's some of the practicalities. First of all, you should know there have been GWASs on everything you can dream of. It's already out there. And the sample sizes are getting larger and larger, which means the predictiveness of them are getting larger and larger. The correlation within ancestry is actually still quite strong, and I'll show you some examples of that. And we are improving in representation, not as fast as I would like, or most people would like, but we are doing a little bit better. And part of it is that there's just so much room to work on improving the interpretation of GWAS and polygenic scores. So this comes from the GWAS catalog. Keep this in mind, there's a bias about ascertainment bias here. This is, you can't really see them here, but these are your chromosomes, 22, and then your sex chromosomes. Each dot, this is the smallest chromosome, by the way, each dot represents a finding from a GWAS that's correlated with that particular location, that particular SNP. All of these different colors are different types of GWASs, not even just one, but like thousands within the yellow. There are thousands of different GWASs within the yellow. The point being, there are so many GWASs and so much work that's already been done. But now you know it's also biased, right? These polygenic scores that are being created from these GWASs predict a fair amount of variance. So it predicts things. This is the polygenic score for education in HRS and Ad Health, and how much does it separate like mean prevalence of college education. You can see that's a pretty big difference between the lowest and highest quintile. And how much is it if you put in that polygenic score? You know, it's higher than household income, much higher than marital status, not as high as parents' education. The thing to know is that we go back to this, European ancestry is always the best place to test this. It'll give you the upper bound of what we have so far. The other ancestry groups are not zero though. It's usually reduced by 50%, sometimes a little bit more. But within the ancestry, it still sorts people based on their predictiveness. You just can't compare across ancestries, right? So within, okay, between, you get into trouble. And there are more and more multi-ethnic and larger non-European studies like Chitwan Valley Family Studies study and LASI, which is in India. So where is this headed? Genomics of behavior desperately needs social scientists. There's expertise on measurement and modeling that I think is important. There's concerns about generalizability, and you know, it's really easy to forget that GWAS is published, that it was published on a biased sample. And it's really easy to be like, oh, it's just biology, it's real. So keeping people remembering where these data were created, a more rigorous interpretation of polygenic scores and what they really mean, and how the social world is probably built into it because of those biases. The other thing is, no matter what, whenever you do any of this work, you always have to fight against and talk about the fact that just because something is correlated does not mean it's biologically determined. And just because something is true at a population level doesn't mean it drives what an individual can do. And then that also gets into eugenics, which makes a lot of people uncomfortable to even talk about it. But this work is being done, and it needs people who are going to remind everyone that we need to be careful of going down those roads again. Okay, so in conclusion, just to get back, there's a lot of potential and a lot of exciting potential. Still a lot of problems with the operationalization right now. But hopefully you can see that there are some practical uses and that this is not going to be going away for any of us. And so learning how to operate around it is probably going to be quite helpful going forward. And with that, I think I'm done. I'm going to stop sharing if that's okay.
Sure, thank you, Coulter. Yeah, I think your presentation was provocative. I'm sure folks have questions. I think my own journey to this, I want to comment on, since you ended with a sort of like, what should we be doing, or should we be doing this? I think all of the evidence that these studies are dominated by European diaspora populations is pretty strong impetus to those of us who study, you know, East Asia, South Asia, Southeast Asia, Africa, Sub-Saharan, et cetera, that we do need more work from us in this area. And I think in contrast to many, at least sociologists, who may view genetic research as anti social science. I actually was going through a little bit of a midlifey relationship with sociology and ready to call it quits when I met Jordan Smoller, who's a psychiatric geneticist. And after some time working with him, I certainly got very energized about what the social sciences have to bring to these questions. In fact, in psychiatric genetics, it turns out that while genetic prediction is there and explains, I think, some of the most severe cases more clearly than more rare cases than mainstream. Things like depression and anxiety and alcohol use are so high prevalence that there's actually a lot of the work looks like what's Coulter described as, okay, let's factor out the genetic aspect that determines these rare cases and see, like, in the general population, what is it actually coming from social circumstances more clearly, which of course was great. And then, the extent to which genetic and medical sciences needed more thorough thinking. Coulter, when I first started going down this path, which I know was like a decade after you started going down this path, I read up on gene environment interaction. And I think I had something like 8 papers where the social environment was summarized with the word cannabis, where they were essentially looking at cannabis exposure as the social environment. And I'm like, yowza, so what happened to the family and the community and intimate relationships and et cetera, And so I really did get much more excited about what the social sciences have to contribute to the future of understanding behavior and health in terms of how the social world that we are used to studying shapes a lot of that and probably shapes how the genetics are fundamentally expressed in some of these kind of outcomes. So that's my 2 cents on trying to go from, a bunch of the limitations that Coulter correctly pointed out to why I actually am excited about and everybody who watches this presentation to do more work in this area.
Well, yeah, thank you, Bill. And yeah, I mean, I was trying to cover things in 35 minutes. I think I actually did. And so there's a lot more to be talked about here, but to your point, Bill, like there's times where I go back and forth between saying, what can genetics bring to social science? And that's a fun discussion to have. There's that other side, what you're saying. And I mentioned a little bit less, which is there's just so much room for social scientists to do really good work in making this more applicable. Because my biggest fear right now is that a lot of the genetics that are being published and discussed have like no social context, no social awareness, even in the creation of their data. And the problem is that once it gets published, it's kind of assumed to be true. And then it's just like, it's a set truth. Now, if you ask any researcher, they're actually far more careful about it, but that's not the way. We all know how literature works. Once it gets formed, it gets shaped, it just becomes this kind of anchor bias. It gets difficult to say like, well, actually, that's not at all what's happening.
Well, I want to hear Sarah's thoughts, and then maybe come back to this discussion about where do we go in the future.
Yeah. So my question is, I mean, you sort of talked about this, but this question of like your ancestry is not the same thing as your ethnicity.
Correct.
But thinking about this in the Chitwan context, like it seems particularly complicated because you have these Tibeto-Burmese groups and then you have these South Asian groups, and I feel like those would have fallen on different ancestry groups in your previous graph. So my question was just sort of about how do we think about this in the CVFS context? Are we always going to be making mistakes by comparing across ancestry groups? Is this really one ancestry group? How do we think about this as an ancestry group? I guess that's
No, that's a great question. I have been able to see at least 1 or 2 graphs of this, of the Chitwan data. And actually my memory, and Bill, you can jump in on this, and I don't want to steal anyone else's thunder. there's a cool thing going on here, which is what we would have thought, that a lot of the things line up in exactly the way that my memory is, like who's related to whom, right? Which groups are more related to one versus another. And it does seem to map on to a lot of those preconceived notions of who should be connected to who, who's closer, genetically closer, who's been marrying within this group and who's not. It's kind of cool how well it maps on, actually. And
yeah, it was a little bit of a complicated cluster graph, Sarah, and Coulter and I, none of us own this. It was screen shared with us from genetics team at Harvard, and so we should request it so we can engage more in this part of the science, which I've kind of, I work with a psychiatric geneticist who's sort of leading that field, so I'm sort of like, okay, I'll do whatever you say, but that was super interesting. And the other part that was kind of, I don't know, Coulter, a little bit scary. So the lab comes back and says, hey, we got, Heather, you might remember exactly the numbers. I'm going to make up a little bit here, but hey, we got 10 cases where the gender that you've written down with that sample doesn't match what we're seeing in the genetic data. And we're a little bit like, oh shoot, a lot of shit happened here. It was stored, it was transported to Kathmandu. It sat in the lab a long time before it was analyzed. It was analyzed, DNA was extracted, it was shipped, so many things that could have gone wrong. I remember there was one situation where rats got out in the lab area and ate like the kits and stuff. So many things could have gone wrong. And I think it might have been Steph who brought this to my attention. I went to Diogenes, should we, what do you think? Should we work with the lab? Should we work with Indra? Should we try and figure out what we wrote down wrong? He said to me, oh, actually, Bill, there's 8 transgender people in the CVFS sample. I'm like, oh, I didn't know that.
Yeah.
He said, there's 8 people who call themselves not what they were born as. And I'm like, oh, didn't occur to me to ask, didn't think about it. In my world, when you get, I think our base here is 10,330 or 40 DNAs, 8 to 10 misses, not bad. But holy crap, out of like the 10 misses, 8 were identified already by them as people. And so at one level, it's sort of remarkably accurate. And I agree with Coulter when I saw it. So, and by the way, let's go critique the social science side of something like ethnicity, right? Yeah. We have taken the diversity of Chitwan, which is one of the few places in Nepal where lots of different ethnic groups intermingle. And we have coded them into essentially 5 groups and a 6th other. So we take the other out and look at these 5 groups, and it's a little bit of a complicated, but it was like you say, like there was a band this way, and then there was a band this way, and a little overlap in this corner, and then another band over here with a little overlap, and you're like, oh yeah, that could totally be true to our sample. And then when you factor in, the Tarai Janajati and the Tarai Janajati have this potential to be much more sort of Burma origin, and the Hill Janjati have this potential to be much more sort of Tibetan origin. But in fact, of course, they coexisted in Nepal for what, 7,000 years we know about. And so who knows, what all mixing actually took place to make you Gurung and Tamang. And, we're taking like Hill Janjati is like Gurung and Tamang and Lamas and this is and that's is all in one group. And probably some of what looks like overlap is because we're not using a detailed enough measure on the social side to see. And I push Coulter a little bit because I still don't know what young and old means in a population sense. In some sense, I had assumed that we all sort of evolved at around the same time. So I went back to this intermarriage versus no intermarriage as a kind of, well, how much mixing is there across populations, which is the way I think about the Nepal situation, where some of these ethnic groups have been much more tightly controlled and not marrying across boundaries than other ones, which I assume, Sarah, sooner or later has got to show up in what we're doing.
Yeah.
And we can come back to the older and younger, because that is a tricky thing, and I'm probably not using the right population genetics terms for that.
Good news. I thought it was funny that you thought I might remember what my high school biology class taught me.
Well, but Sarah, to answer your question more specifically, like how would we do it in Chitwan? It gets a little complicated, but like in theory, yes, if you were to imagine that we are able to sort out using genetics, kind of these major ancestry groups, how they cluster, and there really is a pretty clear like separation of some groups from other groups. And you said, this is an ancestry group, this is a different one, this is a different one. These are admixed, so it's a mixture of a couple different ones. Then yeah, you would use polygenic scores within those ancestry groups. Here's what's interesting though, is, for most of the other studies, like if you're doing one in the United States or in China or Japan or something like that, they might collapse a lot of these groups together and say, this is one ancestry group. So there's almost no way, anytime you study any population, there will always be subgroups that form.
Yeah.
So if you go to Northern Scotland, and there's a study that did exactly this, you go to Northern Scotland, you do genotyping, you can start separating certain clusters into other clusters, which is why if you do a small enough study, you'll separate out into families, which is what you find if you say, go do the PSID and you do this, you just sort of separate it out into families. So, yeah.
And on this question of how it looks in our data, I did want to comment, Coulter, on a couple other topics that you touched on. I think I understand a little bit better for hearing your explanation. Thank you. But this business of, okay, we have 10,300 some genotypes, everything.
Right.
And we'll lock those up, and when we figure out how to use them, we can use them. They have that. And then our team at Harvard is making polygenic risk scores for specific mental health disorders. The first one they delivered was major depressive disorder, the polygenic risk score for major depressive disorder. Turns out that science is evolving continuously at a rapid pace. And one of the things they're evolving is new and better approaches for using the existing however million they have to make a polygenic risk score that might work for your population, even though your population's not in the pool. Right. And Nepal, all of South Asia, I think Coulter was sort of clear in your diagram already on everything genetic, but definitely on psychiatric genetics. There's no South Asia matched DNA phenotype data until this data. So there are no scores calculated for that population. So we're using scores derived from other populations. We used a good version of that. It does predict. I got some questions I could ask Coulter if we have time about, what is all these different ways. They create a polygenic risk score. It's like an integer, but then there's like standardized and corrected and deciles and quintiles and tertiles and but anyway, it does predict in the CVFS population the odds of getting MDD. But Sarah, being delete ethnicity has a much bigger effect, and they appear to be independent of one another. Which I thought, well, that's the first thing that's going to go away. I thought this ethnicity thing is going to go away, but of course, as Coulter explained, not only are there these reasons why it's not the same thing, but the social consequences of being born delete. And it's interesting, right, Coulter, because you're saying, one cool thing about it is the same throughout your life course. Yeah. The cumulative consequences of being delete throughout your life courses are so much bigger than the cumulative consequences of having one gene or another, or at least these genes, I guess, not that extreme version of genes. Right. That yowza, being lifetime delete is depressing, as in most settings, being female is depressing. And, there's a whole bunch of social reasons why that is, that I think the more research they do, the more we'll find it's not genetic. Yeah. Well, and Bill, to your point, you might imagine that you might take the same polygenic score and run it in the, in one of the other groups, and, like Brahmin, and you might say like, oh yeah, it predicts, 5% of the variance, right? It predicts, okay, it predicts some,
and you take it to the delete group, like you were just saying, and you were to say, okay, now we're going to run it there, it may not predict much at all because of exactly what you described, which is, it doesn't matter what genetic predictor you have. The social thing is overwhelming, whatever was there. So you can imagine a situation where that, and that's kind of that example I was giving with the Wisconsin Longitudinal study, right? The big example I usually make is it doesn't matter what genes you have towards alcoholism if the country in which you live does not let you buy any alcohol. Like, it's going to overwhelm your ability to become addicted to alcohol.
No, and Sarah, so interesting in the Nepal data, there's so many restrictions on females getting access to alcohol that there's almost no female AUD in our CVFS data. And it's exactly as Coulter said, it's not necessarily genetic at all. It's just because of these restrictions let me say it a different way. It's because 10% report ever having the opportunity to drink, and like 4% report ever having taken a drink. And then, you get less than 1% have ever had AUD. So that's what it is. No,
and I know we're running out of time, but this is one really cool thing that you could do in Chitwan, right? Is compare men and women, because if you remove the X chromosome, Y chromosome, so any sex chromosomes, there should be no difference at a population level on the polygenic scores, unless there's some mortality selection for anything. And so in theory, they all have the same genetic risk, but because the social structure is so different by that, you could probably really highlight how big of a difference it's making by comparing the genetic effects.
Yeah, I mean, my favorite discoveries, not my favorite, I'm sorry, this sounds horrible. One of the clear discoveries so far is having your parents pick 100% without your input who you're going to marry. It's pretty depressing, and it happens a heck of a lot more to female members of the CVFS than it does to male members of the CVFS. Yeah. Anyway, yeah, it's good. So there's a whole bunch of stuff like that that just shines very clearly against this genetic backdrop of, okay, we have the same polygenic risk score for major depressive disorder handled here.
Yeah, no, it's a really cool opportunity to look at that. Yeah, I know we're short on time. Feel free to email me if you have any questions, or we can come back to this in another lecture sometime.
Yeah, absolutely. We're going to have more and more results to talk about from CVFS for sure. And hopefully that highlights how useful it is in other parts of the world too.
Yeah, exactly.
Great. Thanks, Coulter. Appreciate it.
Thank you,
Bye.
Coulter.
Bye.